Today we are going to talk about neuromuscular junction with updated and modern concepts, especially about the molecular machinery involved in neuromuscular transmission, right? So let's start with that what is neuromuscular junction, right? Neuromuscular junction is basically where the neuronal system connects with the muscular system. For example, in this neuromuscular junction, here what we are talking about, that a nerve ending of the alpha motor neuron, right? When it approaches a nerve fiber, its end become bulbous with multiple buttons, and then on a specific area of extrafusal fibers, cell membrane, they will approach each other. You can say that neuronal end, this is alpha motor neuron, cell body in the spinal cord, and this is axon, and here it is the nerve ending. And this is showing a portion of cell membrane of extrafusal skeletal muscle fiber. Now, where they meet each other, where they kiss each other, but they really don't touch. There's a little cleft in between, right? Where the terminal button, right, meets the sarcolemmal membrane, this meeting point is called neuromuscular junction. Neuromuscular junction, right? Now, this is responsible. What is the purpose of this junction? That this junction will take the action potential from the axon or from the neuron and convert that action potential into a chemical message, and then chemical message will transfer from presynaptic button, right, to the synaptic cleft, and then chemical message will reach to the postsynaptic membrane or this part of skeletal muscle where this nerve ending is joining, and this part of the skeletal muscle fiber membrane or this part of the sarcolemma where this pre-terminal button is coming, this point is called, this is called end plate, motor end plate. So what is here? Here is nerve ending or nerve terminal, and this is motor end plate. This is motor end plate, and this is a very specialized type of a synapse, we can say. It's a very specialized type of synapse, and this consists of basically mainly in three parts. As I mentioned, there is presynaptic region and there is postsynaptic region, right? This presynaptic membrane belongs to the neuron, right? And this postsynaptic folded membrane belongs to the cell membrane or sarcolemmal membrane of the sarcolemma of the skeletal muscle fiber, right? And the gap in between is called synaptic cleft. Actually, this is very ultra small gap. It is just 20 to 50 nanometers, right? And this gap is full of extracellular matrix. But right now, in today's lecture, we will focus on this presynaptic button, how it works, and what are the clinical importance of understanding the concepts related with the events in presynaptic button, right? So let's start with very beginning. That alpha motor neuron or lower motor neuron fires. What I mean by this, that it is giving action potential, and what will happen through the axon, action potential will reach at neuromuscular junction at presynaptic membrane or presynaptic button, right? Now, first of all, we'll talk about what is action potential. Then we'll come to what action potential is especially doing here with the calcium channels, right? Now, The first event is arrival of action potential, right? From which neuron? Lower motor neuron. Don't tell anyone lower motor neurons are lower and upper motor neurons are upper. Lower motor neurons should be defined the neurons which have their cell body in the spinal cord or brain stem. Brain stem also has lower motor neuron. So lower motor neurons are having the cell body in the brainstem or in the spinal cord, gray horn, and lower motor neurons, axons terminate on the skeletal muscle fibers, right? And all other neurons which end up on the lower motor neuron and are coming from higher level, they are upper motor neuron. Because many people confuse, I must make this confusion clear, between the upper motor neuron and lower motor neuron. Many students think it wrongly that lower motor neuron are in spinal cord and upper motor neuron are in brainstem or somewhere else. This is not true. Basically, lower motor neuron should be defined as that it has cell body in the central nervous system and its ending, axon's ending must be on extrafusal fibers of skeletal muscle. These are lower motor neuron. Now, of course, from the spinal cord, gray horn, there are a lot of lower motor neurons going to the many skeletal muscles in the body, right? But what students do not understand sometimes that actually motor neurons in cranial nerves which are going to skeletal muscles. Many cranial nerves have motor neurons which are eventually ending on the skeletal muscles. So those motor neurons which are coming from the brainstem, either they are present in the oculomotor nerve or trochlear nerve, or there is in the fifth cranial nerve. Many cranial nerves have motor neurons. That is not the topic of today. But what I really want to put in your mind, that even in the brainstem, all those neurons which are having their cell bodies in the brainstem or in the spinal cord, and their ends are axonal ends and termination or terminal buttons are ending on extrafusal fibers of skeletal muscles. Such neurons are called lower motor neuron regardless of the fact they are part of spinal cord or they are part of the brainstem. Then what is upper motor neuron? In a simple term, I'm not being very specific, in simple term, that those motor neurons which are coming from a level above the lower motor neuron and influencing the lower motor neuron are called upper motor neuron. So if you are clear about what is lower motor neuron, then it becomes easy to understand what is upper motor neuron, right? So let's start with it, that if, for example, this is a fiber which is taken from muscle biceps, this muscle to flex the arm, then what will happen? That through the brachial plexus, right? Brachial plexus will bring the neuronal fibers. Now what are these fibers? Alpha motor neuron, from the anterior gray horn, from the lower, or you can say C5 and 6, cervical 5 and cervical 6, right? Level of spinal cord, gray horn has alpha motor neurons which pass through the brachial plexus and eventually they innervate through the musculocutaneous nerve to the biceps. And this, in those nerves, this fiber, motor fiber, this is called lower motor neuron. It is also called alpha motor neuron. It is also called alpha motor neuron. Now, when we talk about how this junction work, we'll discuss at two levels. First of all, what are presynaptic events and what are postsynaptic events, right? How presynaptic action potential convert into a chemical message and passes from here and then eventually generates action potential in the muscle, right? Now, let's start with, first of all, arrival of depolarization, arrival of action potential. Now, what is action potential? In a very simple terms, action potential is basically made of two components. For example, nerve membrane has resting membrane potential of minus 70 millivolt. What does it mean? It means interior of the nerve is negative. When membrane is resting, interior of the nerve is negative. Neuron is 70 millivolt, right? And minus 70 millivolt. And here is threshold potential. For example, it is minus 50 millivolt, right? And here is 0. Now what really happens? That when this nerve is excited, alpha motor neuron is excited, it will produce a transient, temporary potential difference across the membrane. Temporary, transient, reversible, propagating. These are the features. There is temporary or transient. Yes. Temporary, transient, propagated, potential reversal across the membrane. That if this is the membrane, let's suppose if this is the membrane, then across the membrane potential reverses, right, temporarily, and then it propagates throughout. I will not go into detail, but just let me tell you, when there is no impulse coming, it is minus 70 millivolt. What really happens when this is excited, right? It activates the voltage gated sodium channels. When voltage gated sodium channels open, lot of sodium will go in, because you know extracellular sodium is more and intracellular sodium is less. Why it is so? All of you know that their sodium potassium ATPase is pump, and these are always taking sodium out and bringing the potassium in. So extracellular environment is rich in sodium and intracellular environment is rich in potassium. So what really happens? That when it is properly excited, this neuron membrane is excited, right? Then voltage sensitive sodium channels open. When voltage sensitive sodium channel open, naturally sodium will move from high concentration to the low concentration. So very rapidly there will be influx of sodium. I'm going to explain action potential first of all, arrival of action potential. When action potential is generated in alpha motor neuron is stimulated in such a way that initially its potential, few cations or positive charges enter in, right, at this level. Soma or dendrite, few cations enter it. When these cations enter due to some stimulation, what will happen? That which was minus 70, it will become less minus, it will become minus 60, and it will become minus 50. So up to this point, if potential can be exchanged from resting membrane potential by putting some cations in the neuron, and it becomes less negative, it is going towards the positive, less negative, so much so that it touches the threshold potential. What is threshold potential? Threshold potential is that voltage or that potential at which voltage gated sodium channels open. Voltage sensitive sodium channels open. Now, suppose it was minus 70 millivolt here, and once it was stimulated, whatever the cause of stimulation, for example, that must be some neurotransmitter which stimulated the alpha motor neuron, that will trickle some cations in. Those cations or positive charges will take the resting membrane potential from minus 50. When positive cation will go in, they will partially neutralize the negativity, and it will become less negative from minus 70 to minus 50. If it reaches minus 50, automatically voltage gated sodium channel will open. Whenever automatically voltage gated sodium channels will open, sodium will go in. As sodium will go in, everywhere it will become membrane will become minus 70 to minus 50. Here, the sodium channels open. When voltage gated sodium channel open, too much sodium very rapidly comes in, and too much sodium enter into axon, enter into cell, right? When enter into cell, this electronegativity is lost and potential moves towards positivity. Maybe it become positive 20 millivolt, right? So what we will say, when alpha motor neurons are stimulated, right? During this stimulation, stimulating factor takes the resting membrane potential to the threshold potential. Threshold potential is the potential at which automatically, simultaneously, voltage sensitive, to be more accurately, threshold voltage sensitive sodium channels will open. And when this will open, as you know, sodium is too much outside the cell, it will rapidly go down its gradient, and there will be heavy influx of sodium. So if I make this neuron, this part of the neuron here, right, there will be, these are supposed sodium channels. Let's suppose here from minus 70 it goes to minus 50. When it will open up, lot of sodium comes in. Now from here the story will go automatically. When a lot of sodium will come in, it becomes plus 20. Do you think it is now polarized negatively? No. Originally, when it was resting, it was negatively polarized. And once heavy sodium influx occur due to opening of the voltage gated channels, then negative polarity is lost. When negative polarity is lost, we say that there is depolarization. The negative polarity is no more there. It is no more minus 70, it is going towards 0. When it is approaching or hovering around 0, we say membrane which was negatively polarized, resting membrane potential, right, negatively polarized, it has become, this membrane is now depolarized. Now remember this thing, once one part of the membrane becomes depolarized, lot of sodium comes. If this part of the membrane becomes depolarized, this sodium which comes in, it will go to nearby area. All areas, you know, membrane, resting membrane is what? Minus 70. This sodium will float inside, and this positive cations coming in will convert the next group of sodium channels by changing the voltage here. Because whatever positive came from here, cations, in the exoplasm, they will go to the neighboring areas. And in the neighboring area, when this positive cations will come, neighboring area resting membrane potential will automatically achieve threshold. And when it will achieve threshold, then booze, another group of sodium channels open. Lot of sodium again come in the second area. Now this second area, when lot of sodium is coming in, sodium spreads on the sides, second area is depolarized, and it also takes the neighboring area to the threshold, and the neighboring area undergo depolarization due to opening of the voltage gated sodium channel. So what is happening? I'm telling how the action potential arrives here. Action potential consists of two main components. There are other components also, two main components. Then membrane first depolarizes, and this wave of depolarization moves along the axon. So it means this wave of depolarization is what? It is propagated. It is propagated, right? Why it is propagated? Because whatever heavy sodium comes here, and whatever the patch of the membrane become depolarized, it will make the adjacent area, the resting membrane, to threshold, and then adjacent area will depolarize. Second area will depolarize. Then second area of depolarization will take next area to threshold, and then this will go to depolarize, so and so forth. What will happen? That once voltage gated sodium channel start opening, they keep on depolarizing the membrane, and this wave of depolarization sweeps on the membrane up to the terminal. Wave of depolarization. What we can say, if we are inside the axon, if I'm standing inside the axon, right, what happens? Here it is electronegativity. But when wave of depolarization comes, then sodium is coming there. So what we can say that actually, number one, we can say wave of depolarization is coming. We can also say that wave of loss of negative polarity is coming. Wave of loss of negative polarity is coming, or wave of sodium influx is coming. So what is depolarization? Depolarization is when membrane negative internal side polarity is depolarized, lost. Right? Why it occur? Due to potential in the membrane has reached from resting membrane potential to threshold potential, and at threshold voltage gated channel open. And once in one patch of axon opens the sodium channels, voltage gated sodium channel, so much sodium will go in that adjacent area resting membrane will go to threshold. And then again their depolarization will occur. And then next area, so and so forth, wave of depolarization goes. But there is one thing. As wave of depolarization is advancing, for example, first there was depolarization in area number 1, then area number 2, and let's suppose eventually this led to the area number 3. So what we can say, wave of depolarization or wave of electropositivity inside or wave of loss of electronegativity inside is moving. And if you are standing here, right here you are with your beautiful nose. If you are standing here and you look on the axon, you see there are, let's suppose, sodium ions are colored blue, hypothetically. You will see that these are blue men. So blue men are jumping in, right? And membrane is undergoing process of depolarization. But there is another thing. Whenever one patch undergoes depolarization, it makes the next area to threshold and depolarization, and itself, it reverses back. It means it repolarizes. Now, why it was depolarized? Because cations came in, positive charges came in. Actually, the moment this area becomes depolarized, next area will taken to threshold. So it is ready for depolarization, but within this area, there are, for simplicity I'm saying, there are depolarization sensitive potassium channels. There are depolarization sensitive potassium channels. Or we can say that when membrane go near the depolarization, these are also voltage sensitive potassium channel. The difference is that sodium channels open when resting membrane potential from -70 goes to -50, suppose. Then they are voltage gated sodium channel open and sodium moves from out to in. But once a part of a membrane becomes depolarized, within milliseconds, even once sodium come in, within milliseconds, or in a very brief time, I must say, as soon as this part of patch of the membrane get depolarized, in the same membrane, voltage sensitive or depolarization sensitive potassium channels will open, and cells are bags of potassium. Cells are loaded with potassium. So once potassium channels open, potassium will go out. So potassium is also positively charged. So sodium came in and wave of depolarization started, right? There was gain of positive cations and resting membrane potential went towards zero potential, loss of polarity, depolarization. But the moment patch is depolarized, potassium channels open and inside the potassium will move out. And when more and more potassium will move out, what will happen? If it was plus 20, some potassium moves out, it will become progressively negative, right? So this component which is showing that membrane potential from peak potential is going back to the resting membrane potential, this is called repolarization, because membrane is again polarized. Repolarization. again polarized negatively. So what happens? That when alpha motor neuron is stimulated, there is a wave of depolarization followed by a wave of repolarization, right? Or we can say the wave of positive cations influx followed by wave of positive cations efflux. More accurately, we can also say there is a wave of depolarization or wave of sodium influx or cationic influx which take the resting membrane potential towards zero potential and membrane become depolarized. The moment depolarization progresses ahead, the membrane which remains behind, their voltage gated potassium channels are activated and potassium efflux will occur. So we got the cations, we lost the cations. By gaining the cations inside, we made it from -70 to eventually +20. And by loss of cations, again we re-established negative polarity and eventually we reached to -70. Now, because wave of depolarization, right, is followed by wave of repolarization, we can say that both of them comprise a potential, fluctuating potential across the membrane which is in action, fluctuating potential across the membrane which is propagating, which is in action. So depolarization plus repolarization, both are moving. First, what is moving ahead? Wave of depolarization, followed by wave of repolarization. So both of them, when they are in action, we say the sum of depolarization, repolarization, and some more changes which I will not mention here, all those changes are called action potential. So what is action potential? When we say, let's come to our true business today, presynaptic button, right? Or at presynaptic button, when we say action potential arrives here, who arrives here? Wave of depolarization arrives here, followed by the wave of repolarization. And when depolarization followed by repolarization phenomenon is seen, we say, what is arriving? Action potential. So what is action potential? Action potential is the process in which first membrane undergoes depolarization due to sodium influx and immediately after that undergoes repolarization by potassium efflux. And this transient electropositivity and then again reversal of electronegativity inside the membrane, right? This potential fluctuation is called action potential. So once the action potential come here, it is worth repeating, once I say action potential arrives here at the terminal button, what has arrived? Wave of sodium influx has arrived here. Of course, it will be followed by wave of potassium efflux. Once wave of sodium influx come here, or wave of depolarization come here, the membrane here, when it is depolarized due to sodium influx, it will become, yes, tell me, plus 20 millivolt or plus 10 millivolt or even 0. But the important thing is it was negatively polarized and heavy sodium voltage gated sodium influx leads to loss of that negative polarity. So when this wave of electropositivity inside the membrane arrives at the terminal button, here are someone waiting. Who is waiting? Calcium channels. There are very special type of calcium channel. We call them PQ channels. What do we call them? PQ type calcium channels. Now, these are PQ type calcium channels. These are a very special type of voltage sensitive calcium channel. There are many types of calcium channels. Some calcium channels are operated by different ligands, right? But these are voltage sensitive potassium channels. To be more accurate, calcium channels, yes, voltage sensitive calcium channels. To be more specific, I must say, they are action potential sensitive. Action potential sensitive calcium channels. To be more specific, they are depolarization sensitive calcium channels. So, once action potential arrives here, this is an electrical phenomenon, and this, once it reaches terminal button at presynaptic membrane of neuromuscular junction, it finds itself where? It finds itself approaching voltage sensitive PQ type calcium channels. And those calcium channels, they are so sensitive to depolarization. The moment depolarization comes, right, depolarization is coming like there are sparks of sodium in flux. The moment this electropositivity comes here, these channels will open, and you must be knowing that extracellular calcium concentration is very high as compared to, don't say intracellular, say intracytosolic. Remember, in extracellular fluid, calcium concentration is very high. In the cytosol, calcium concentration is very low. But in the cytoplasm, mitochondria and sarcoplasmic reticulum and so some other organelles are very rich in calcium. So don't talk about extracellular, intracellular. More accurate will be extracellular fluid and cytosolic fluid, right? So what happens, as soon as this channel, which are opening outside in the extracellular fluid and inside they are opening into cytosol, so as potassium concentration outside is more and inside is less, potassium will very rapidly pass through this, right? And calcium, yes. And in this way, this terminal button will have a transient on arrival of action potential or on arrival of depolarization in terminal button PQ calcium channels, which are voltage sensitive, they will open up and they will give a puff of calcium inside. They will give a puff of calcium, a small amount of calcium will go in. Now this calcium which comes in, right, this is going to move the neurotransmitter from these vesicles to the membrane and unloading the neurotransmitter here. Neurotransmitter here, now we will talk about it. That from where these vesicles come and what kind of neurotransmitters are here and how calcium alters the behavior of these vesicles. Now, vesicles are basically, listen carefully, cannot be synthesized here. Vesicles are synthesized in the cell body in the central nervous system, because formation of vesicle and all other proteins associated with vesicles or any other proteins which are present in this membrane or protein or channel transporters present in vesicular membrane or axonal membrane or terminal button membrane, all these proteins are made in the soma, because formation of protein requires RNA, RNA requires DNA, and DNA is present in, yes, in the nucleus of the soma. So actually, vesicles and other proteins which are present in this terminal button, they are basically, originally they were synthesized in the cell body under the direction of the genetic material, and then something called exoplasmic flow, right? So these components will flow along in a very controlled manner, right? These vesicles along with special cytoskeleton inside, they will be transported to the terminal button. So vesicles are coming from soma. These vesicles are very important. We should not lose these vesicles. And if we lose, we should recapture these vesicles. I will tell you later. Now, these vesicles, they are full of a neurotransmitter called acetylcholine. So they are loaded with acetylcholine. Let's suppose that here is the vesicle, and this vesicle is loaded with, for example, this is the terminal button. I'm just showing the part of this here, and here is the vesicle. Here were filaments which brought these vesicles from the soma, right, for the trafficking. Now this vesicle is here. Now, how acetylcholine comes here? Suppose this is acetylcholine. How acetylcholine reaches here? First of all, remember acetylcholine has two components. Acetylcholine has two components. One component is choline. Suppose this is choline and this component is acetate. Choline and acetate. And both of them together are called acetylcholine. Now, from where choline comes, from where acetate comes, who makes them fuse together, and who concentrate this newly formed combination acetylcholine into vesicle. Let's talk about it. First of all, from where the choline comes. Actually, nerve ending cannot synthesize choline itself. It does not have any synthetic machinery for the choline. So what it will do? It will capture the choline from extracellular environment. Actually, there are special transporters here. What is this membrane? Neurolima. Neurolima or terminal button membrane. I'm talking about here. There are transporters here. These are special type of protein. Now, what this transporter is doing? This transporter is actually choline uptake mechanism. Whatever choline, these transporters are present in millions of numbers here, and wherever they in extracellular fluid or in the cleft, synaptic cleft, wherever they find the choline, they transport inside. So these are what? Choline transporters. So from where the choline comes? Choline is captured by special transporter proteins, choline transporter type 1, right? They take the choline from the extracellular environment. But remember, ultimate source of choline, major choline, ultimate source is our diet. Our diet, from there choline is absorbed and then it goes to the blood and extracellular fluid, and there are many functions of choline. One of the functions is that this choline will be captured by the cholinergic nerve endings and it will be concentrated inside. It will be taken up and concentrated here. So choline comes here. This is the choline, right? So what is this transporter called? Choline transporter type 1. Now once choline comes in, what happens? Now we have to find acetate from somewhere. Luckily, this nerve ending have its own acetyl-CoA. Why? Because it has mitochondria. Here are mitochondria, the powerhouses, right? And from the metabolism, they are finding acetyl-CoA. So actually in the nerve ending, mitochondria, right, they are providing the acetyl-CoA, right? Let's suppose acetyl-CoA, we show it like this. Now, we have acetyl-CoA here and we have choline here. And you know, there is someone who requests both of them make a union, right? So someone motivates, what is this? Acetyl-CoA and choline. Acetyl-CoA and choline. They fuse together under the influence of an enzyme, right? Here is choline, and when the choline and acetyl-CoA, they fuse with each other, the acetate component of the acetyl-CoA fuses with the choline, and this compound is now called acetylcholine. Choline which is acetylated, acetylcholine. And those nerve endings which are having acetylcholine, we call them cholinergic nerve endings. What we call them? Cholinergic nerve endings. Now, at neuromuscular junction, the nerve ending of alpha motor neurons are basically cholinergic nerve ending when we talk about their neurotransmitter type. Now, choline was taken from the extracellular fluid and a lot of it was recaptured, as I will explain later, from the cleft. And once choline comes in, the mitochondria provide acetyl-CoA, and under the influence of an enzyme, yes, this enzyme, right? Under the influence of an enzyme, both of them fuse together. So what is the function of this enzyme? The function of this enzyme is that it is transferring the, what is this? Acetate to the choline and resulting into acetylcholine. So what should be the name of this enzyme? Choline acetyl transferase. What is the name of this enzyme? This enzyme catches the choline and take an acetate group and transfer to choline. So we call it choline acetyl transferase, right? So it means here is the enzyme. What is the name of this enzyme? Choline acetyl transferase. Choline acetyl transferase. So from here you will tell me what came in? Choline. Right? And from mitochondria what came? Acetyl-CoA. And enzyme worked on both of them, and both of them eventually converted into acetylcholine. Now, this newly synthesized acetylcholine is present in the cytosol. This is very valuable substance here. It is a real chemical message. And if it is so important, it's so valuable, it should not be left here. It should be properly packaged. So, from the soma, previously, the vesicles which have come here, those vesicles know whenever they know that there is acetylcholine in cytosol, as soon as it is synthesized, they will capture and uptake it. It means that there must be another type of transporter which is present over here. Another type of transporter which is present over here. Function of this transporter is that it will take acetylcholine and transfer and concentrate inside the vesicle, right? And this transporter is called vesicular acetylcholine transporter. Now this was just choline transporter, which was in the outer membrane, or you can say which was part of the terminal button membrane, right? Or part of the presynaptic membrane, right, in the cholinergic nerve ending, right? So this transporter is choline transporter, and this transporter is vesicular acetylcholine transporter. The difference is that both of them are transporters, but this transports choline and this transport acetylcholine, and between them there is someone who makes the choline and acetate fuse with each other, and this is called choline acetyl transferase. In this way, in every vesicle, newly synthesized acetylcholine is being concentrated. You know, one vesicle might have up to 10,000 molecules of acetylcholine. And now we can say that these clear vesicles which are present in the presynaptic button, these vesicles are loaded with acetylcholine, right, which was synthesized. This acetylcholine was synthesized within this terminal button. This acetylcholine was not synthesized in the soma. It was synthesized locally. So it means terminal button was working as a cholinergic factory, cholinergic synthesizer, and acetylcholine synthesizer and acetylcholine storehouse. So this is what we can say. One of the function of the terminal nerve ending is that it works as acetylcholine synthesizer and in the vesicle acetylcholine storehouse. So we can say that nerve ending, cholinergic nerve ending, work as a factory and packaging unit of acetylcholine. Now, what we have? We have a lot of vesicles here. And these vesicles are loaded with, every vesicle is loaded with about 10,000 or so molecules of acetylcholine. Now, when action potential will come, or wave of depolarization will come, if I miss many steps, I will just say that acetylcholine, these vesicles fuse with the membrane and acetylcholine is released out. It comes over here, acetylcholine, and then it will work with special nicotinic cholinergic receptors. But no, I don't teach like that. I will tell you all the steps, all the molecular machinery, and what are the clinical importance of that, right? Let's start with it. We were talking about, now we know that in the nerve ending, we already have vesicles and they're loaded with acetylcholine. And we also know that when alpha motor neuron is stimulated, action potential propagates along the axon and eventually reaches to the nerve terminal, right? This arrival of action potential leads to opening of voltage sensitive calcium channels, PQ type calcium channels, and calcium influx occur. Now, this calcium will come in, and eventually, once this puff of calcium comes in, release of acetylcholine occur. All this process is called exocytosis. Exo means outside, cytosis means cell, throwing something outside, right? Now, how exactly it occurs? What are the molecular machinery for this? How calcium can do this? Calcium is not like a pump that calcium will come and push the vesicle towards the membrane. Calcium will just give signal, only this signal, that action potential has arrived. Calcium influx means action potential has arrived. Calcium influx means there is a message, release acetylcholine. There is a message that vesicles should move towards the synaptic membrane, presynaptic membrane, and fuse with it and release its content in the synaptic cleft so that it comes here. But once calcium comes in, what it is doing exactly? Actually, in these vesicles, this is the vesicle, right? Loaded with, what was it? Acetylcholine, right? This vesicle has some special type of, you can say, special legs, right? Whenever this vesicle, now listen, if these vesicles have these special legs, whenever this vesicle will come near this membrane, do you think it can bounce back or fuse with the membrane? Answer is, it will just bump. So you say, if I'm that vesicle, my arms and legs are like that. And I cannot sit anywhere because my legs and hands are pushing the things. This protein is also like pushing protein. It is all the time pushing the things away, right? So vesicle cannot naturally, without any stimulus, fuse with this membrane. Vesicular membrane, without any stimulus, cannot fuse with terminal button membrane, right? So vesicle remain inside the button, but rarely one or two vesicle may fuse, but not in bulk, because when action potential comes, usually 200 to 300 vesicles fuse with one action potential. But if there is no action potential, maybe some random bouncing occur and by chance one will release its content, right? Now what calcium is doing? Remember, there is not only these, first of all, name of this protein. Name of this is synaptotagmin, because these synaptic vesicles are tagged with these proteins. These synaptic vesicles are tagged with these proteins, so these are called synaptotagmin. Why I am talking so much about synaptotagmin? Because as long as no stimulus is here, no calcium is here, these synaptotagmin are in a very active state and do not allow vesicle to fuse with other membrane. But the moment calcium come, right, calcium will bind, this calcium which is coming in, calcium will bind with synaptotegmin. And what will happen? Synaptotegmin will undergo conformational change. Now this area of the vesicle can hit here, and this area of the vesicle has already some proteins, and these proteins, okay, I will show you more accurately. These proteins are just like one part of a zipper. These are like proteins like this. Now, as long as there is no calcium, they will not allow these proteins of the vesicles to interact with the membrane protein, which are also similar like that. These are membrane protein. Now, these are membrane protein, and you can imagine these are vesicular proteins. But as long as this naughty, this is there, can this group of protein fuse with this group of protein? No. But the moment calcium come here, calcium bind here, calcium binds here, calcium binds here, these synaptotegmins are pulled away. When they are pulled away, then these vesicular proteins, this vesicular protein, right, this will interact with these membrane, axonal membrane or terminal button membrane proteins. These proteins and these proteins will go near to each other, and they will coil around each other. They will hug each other. They will love each other. You know, the bad thing has gone due to calcium influx. So you can say these were vesicular breaks. When calcium comes, these breaks fail. Right? You can say in some very primitive cultures, brothers become very angry if sister goes and meet her beloved. They don't allow the sister to go and fuse. Right? These are those brothers. These are those super traditional brothers, and they don't allow the vesicle to fuse with the membrane. But if calcium comes, something happens, they go away. Now sister will move towards its boyfriend, and she has already some system which can interact with the boyfriend. Now what are these proteins and what are these proteins? Because these proteins are helping to fuse the vesicular membrane with the terminal button membrane. Actually, these proteins are called snare proteins. What are these proteins called? Snare. Snare proteins. Now, snare proteins, some proteins are on the vesicle, so they are vesicular snares, and some are on the target membrane, so T snares. Again, I will repeat it. Vesicles, by virtue of presence of vesicular snares, they love to hug the what? Target snares. But the problem is this synaptotegmin. Function of the calcium is remove the or change the configuration of synaptotegmin. When synaptotegmin is away or molded in such a way that now vesicular snares can interact with the, what is this? Target membrane snares, right? Now, these snare proteins, classically, there are many, but most famous among all of them, which is vesicular snare, the most commonly known is synaptobravin. Synaptobravin. You know, this needs to be brave girlfriend. Go and see the boyfriend. Synaptobravin. So what happens if you remove these naughty proteins, brotherly proteins, we give them calcium, and they become themselves inhibited, and they are altered in their configuration. And once they, by calcium, synaptotegmin are altered in such a way that they are no more able to allow the snare proteins of vesicle to prevent the meeting of snare proteins of target membrane. So, calcium has only one function. Calcium comes in, interact with synaptotagmin and activate it in such a manner that it is unable to block the snare proteins. And then what happens? These snare proteins, look here. One snare protein group, what are these? Synaptobrevins. Synaptobrevins. And of course this is vesicular protein. And they are waiting here. What are these? These are basically, this is vesicular, this is target snares. Target snares are basically two types which are very well known. One is syntaxin, and other is SNAP25, right? So what we can say that now I will make it here. This is the vesicle. Wow, this was cholinergic vesicle. In the cholinergic nerve ending, they were having synaptotagmin, and they were also having some other proteins. One of them we already mentioned, that was acetylcholine transporter. But there is part of some of the fusion proteins are there. One is what? Synaptobrevin, which is also called vesicular snares, and on the target membrane where they need to fuse, there are T snares, right? And two of them I want you to remember: syntaxin and SNAP25. So when these are away, they are not disturbing interaction of them, this will move towards it. And then once vesicles come over here, right, then vesicular snares will interact with T snares. When they will interact with each other, right, they will coil and pull this membrane towards this membrane. And you know this membrane is also lipid, this membrane is also lipid. So when this will touch this membrane, it will fuse. And when both membranes will fuse, I mean next stage will be, it will touch over here, very happy. And you know, happy people lose something. So it will lose the acetylcholine. And process of exocytosis is complete. The release of acetylcholine in synaptic cleft. Right? Now these are electrical and molecular events which occur presynaptically, right? I will repeat them 1, 2, 3 manner. Number 1, yes, arrival of action potential. Number 2, opening of PQ type calcium channels. Number 3, calcium influx. Number 4, synaptotagmin is altered in such a way that it does not impair the formation of snare complex. Then we already know the vesicles were imported from the cell body, from central nervous system, and acetylcholine was actively transported from the cytosol. In the cytosol, there was an enzyme which was fusing the acetyl-CoA and choline, which was taken from extracellularly, and acetylcholine, whatever was made here, was concentrated here. Now, once synaptotagmin receives what? Calcium, it alters and allows the vesicular snares and target membrane snares to interact with each other. It means arrival of calcium removes the synaptobrevin related inhibition of the infusion. Synaptotagmin, calcium bind with synaptotagmin. Remember, synaptotagmin is not the part of snare proteins. Once synaptotagmin is under the influence of calcium, it no more inhibits the interaction between V snares and T snares. V snare, synaptobrevin; T snares, syntaxin and SNAP25. They interact with each other, they coil with each other and force the two membranes to fuse with each other. When these membranes fuse with each other, what really happens? Acetylcholine is released into synaptic cleft. Now here, I want to tell you something. when this vesicle fuses with membrane, if with every action potential 2 to 300 vesicles are fusing, and if 1 million action potential come, so many hundreds of millions of vesicles fuse, and vesicles are made of membranes. And if vesicles keep on arriving and fusing, what will happen? Two things will happen. Number one, this membrane enlarged, and nerve ending will become very bulbous and dysfunctional. Number one. Number two, there will be deficiency of whole apparatus of vesicular system. So actually, once vesicular membrane becomes the part of, what is this? When it fuses, it becomes the part of, for example, here it is fuses, right? So it becomes the part of, what is this? Terminal button membrane. This membrane fuses with this membrane. Then this membrane is very specialized because it was having some transporters, it was having synaptoptegmin, it was having vesicular snare proteins. All these things are very valuable because we cannot synthesize them here. They are only synthesized in the soma, and we cannot allow this very specialized piece of vesicular membrane to be lost. So once it has released its content, then we have to pull back. We have to pull back the vesicular membrane inside. For pulling back, there are special type of proteins which will come and bind here. When vesicular membrane becomes the part of the nerve ending membrane, this specialized vesicular membrane will attract certain proteins which will come and bind. And they will pull this membrane inward. And membrane will again start going backward, but now it is empty. And eventually it will go in. Empty vesicle. Now these proteins are called clathrins. What are these called? Clathrins. So function of clathrin is to reclaim the vesicular membrane with its all specialized proteins. But now, once it is reclaiming, right? What are these? Yes, please. Clathrins, very good. Now, another special type of protein comes here, at this place, and it pinches this away. This protein which came here and pinch the vesicular membrane away, act like a scissor, this is called dynamin. So acetylcholine will be released in synaptic cleft, and vesicular membrane with the specialized proteins will be pulled back and reclaimed, and it will be again filled by arrival of choline, whatever, right, and acetyl-CoA, and whole process will repeat. In this way, these vesicles are locally synthesizing acetylcholine, releasing acetylcholine, and reclaiming the vesicular membrane, and again synthesizing acetylcholine and filling these vesicles. Right? Now, few more points. There is some areas of this terminal button, right? Some areas are called, these areas are called active zones. What are it called? Active zones. There is some special activity going in some areas of terminal button membrane, active zones. These active zones, if you go to the postsynaptic membrane, that is also very much folded to increase its surface area. Now, presynaptic, this is the crest area, this is the trough area. Now, this crest area has cholinergic receptors. This crest area is cholinergic receptors. Now, this opposite to this crest area, there is specialized thickening of the membrane and some area. Now, this area and this area. This is called active zone, which is exactly opposite to the crest of the postsynaptic membrane, where there is concentration of cholinergic receptors, nicotinic, muscular, cholinergic receptors. Now, this active zone has some speciality. at a super micro level, if you go, these active zones are very efficient. Why? They have especially high concentration of calcium channels. Along with that, they have high concentration of, what is this? Vesicles, high concentration of synaptotagmin and associated V and T snare proteins. So it means whole vesicular functional complex along with the calcium channels, they are concentrated in different patches of the presynaptic membrane. Which part of the presynaptic membrane? That part of the presynaptic membrane which is just opposite to the crest of the folds of postsynaptic membrane. This enhances the biological efficiency of the system, that acetylcholine is released near the crest where it will rapidly diffuse and stimulate the target cholinergic, nicotinic, muscular receptors, which are basically cationic channels. We will discuss later. Now, in next section, we'll talk about clinical correlates related with, what is this? Presynaptic button. We'll talk about what drugs and toxins act on the sodium channels and impair the arrival of action potential. We will talk about the diseases of these calcium channels, or what drugs can block these calcium channels and produce weakness because transmission will not occur. We will talk about if congenitally this enzyme is defective. We will talk about what are the toxins which can destroy the snare proteins and they cannot fuse with each other, and many more clinical aspects. Right? Let's have a break. We'll continue later with the clinical aspects which affect the presynaptic membrane in such a way that transmission from the presynaptic side becomes dysfunction. Thank you very much for today.